This video is the third video for photosynthesis in Unit 2. Today we'll be looking at the Calvin cycle and also something called photorespiration, which is what the plant has to do if there is no CO2 available. So we'll look at that. Just a quick review. Here's the light-dependent reaction. ATP and NADPH are made. Electron carrier NADPH is carrying the electrons that were provided by water, and ATP was made through the passage of protons across the membrane, across ATP, and so those two molecules are going to be carried into the Calvin cycle now using CO2. Again, CO2, an inorganic molecule, is going to be used by an organic system, so that idea is called carbon fixation, making an unusable molecule usable, and it's going to be put at energy. ATP and NADPH are going to be put into a sugar. The Calvin cycle happens in a series of steps. Here they are. As with normal way of doing things, I'm going to make a separate drawing of this, but here's the picture if you want to see kind of the big picture of this before we get into the weeds. And so Calvin cycle takes place in the stroma of the chloroplast, and we're going to do 3 CO2s at a time for this. And this is a cycle, so that it kind of is like imagine a merry-go-round. You get on it at one point, get off at another point, but it's just continuing the circle. And so CO2s are going to get on, sugar is ultimately going to get off. Those CO2s are going to be converted, but the other innards of the cycle don't really change a whole lot. And so at the end of this cycle is a 5-carbon molecule. Not important to know the name of that 5-carbon molecule. It's called ribulose biphosphate. You really need to know, but no need to know. RUBP. I will just write RUBP in order to refer to it. And it has 5 carbons. I draw the 5 circles to represent those 5 carbons. And there are 3 of these, just like there are 3 of our CO2s. Now each CO2 is going to be put onto that RUBP molecule, and there's an enzyme that's going to do that. That enzyme is called ribisco. Ribisco is one of the most abundant organic compounds in the world. It does this plants all over the world. There are lots of plants and lots of cells in those plants, and so you can imagine a big amount of this is going to be present. Each one of those 5 carbon molecules is going to have a single carbon attached to it, and so which would you expect to have 3 6-carbon molecules created. Now this 6-carbon molecule that is created is an unstable molecule. It is going to quickly break apart. Each one of them is going to break apart, and so now you're going to have 6 3-carbon molecules. These are called phosphoglycerate, and we'll just call them PGA for short. Now at this point, these molecules are ready to be manipulated. They're not going to have any more carbons added to them, but they're going to be energized. And so I'm going to draw a little thing around it to just show that it's been energized. And the way that these molecules are going to be energized is twofold, and we should know what that is. They're going to have 2 electrons added to them, essentially. That is going to happen with 6 NADPH. Remember, the NADPH is going to come in, add those electrons. When they are used up, they're going to be 6 NADP plus, and those 6 NADP plus are going back to the light reaction to scoop up more electrons. What next? Well, we have 6 ATPs, and those 6 ATPs, each one's going to phosphorylize a PGA molecule, giving a phosphate to it. turning those into ADPs, and those are headed back to get some more phosphates from the light-dependent reaction. Remember the cycle here. These molecules are just circling in and out. And so at the end of this, we're going to have 6 molecules that are known as G3P, or glyceraldehyde 3-phosphate. G3P is more than sufficient. And so these are going to circle around here, and this is where something's going to get off the train. That's something that's going to get off the train is one of those G3P molecules. And what we're left over with is 5 G3Ps, 5 3-carbon molecules. Now, we took 3 CO2s, converted that into one 3-carbon molecule called a G3P. This G3P can be used for a number of things for the cell. It can be used to build things. It can be used to store energy. If you take 2 of these G3Ps, and you have a 6-carbon molecule called glucose. And so this is where we get to glucose in the Calvin cycle. So glucose is then stored up, usually chained together to make starches, and stored in a plant. But those G3Ps again can be used to make any organic molecule, really, because remember, carbon is the backbone of all of those organic molecules. And so right here, we really just have a rearrangement that needs to take place. We have 5 3-carbon molecules, and we need to get back to 3 5-carbon molecules. So we have the same number of carbons here between these two. We just need to rearrange them. In order to rearrange things in the cell, we need energy, break bonds, form bonds, and so it's going to require a little bit of an input of energy to do that. And so 3 ATPs are going to come in, be used to rearrange those molecules so that the system returns back to where it began, so that more carbon, more CO2s can get on the Calvin cycle, make more sugar, and the rest is history. So, what does a plant do when it's unable to do this? Well, let's look at a situation. Transpiration is the idea of water leaving the leaves of a plant. This is a normal and healthy thing that plants do. Water leaves the leaves of the plant, thus pulling water up from the ground via that capillary action that we've talked about. And in a normal environment where water is plentiful, this is not a problem. You take like the area that we live in here in the western part of Kentucky. There's plenty of water. The trees do well. There's never a need for water conservation unless some sort of drought or something. For the most part, there's healthy. But there are other parts of the world where sunshine is abundant. Transpiration would also be abundant, but the problem is water is not abundant. And this is a problem for plants because these little holes called stomata that are in the plants, that's how water leaves. But that's also how, as you can see here from this picture, how CO2 comes in. CO2, in order to do the Calvin cycle, comes into the leaves the same place that water leaves the leaves. And so if the stomata of a plant, say in the desert, has to remain shut in order to conserve water, there's also going to be a problem where that plant is unable to do the Calvin cycle. With plants that are in this area of the country, like that are just kind of doing this normal system, those plants, if there's no carbon available to do the Calvin cycle, will go through a process called photorespiration. Photorespiration is essentially doing the Calvin cycle using oxygen instead of CO2. And so what happens? Well, oxygen is used instead of CO2 here, and so there's no carbons put into the system. And so if you start out with these 3 5-carbon RUBPs, that's 15 total carbons, right? Well, if the Calvin cycle happens and no carbons are put in, how many carbons are you going to have left? Well, the Calvin cycle takes out 3, so you're going to have 12. Do it again, 9. As you can see, this is not something that the plant can sustain. Plants in this part of the country and in parts of the world that don't have a lot of need for water conservation, those plants are called C3 plants because they deal in this 3-carbon system in which they're taking 3 carbons to do the Calvin cycle. In other parts of the world where sunshine is abundant, water is not so abundant, there are 2 other methods that can be used. And that brings us to these types of plants called C4 plants. There's a real simple way of looking at this, and again, we can get pretty complicated here. I'm not even going to draw a picture for this, but we're not going to get complicated. Here, just notice a few things that you immediately recognize. CO2 coming in. You have the Calvin cycle down here to make sure. Simple kind of thing. But notice in the interim, there's this little system. And this system is there to prevent photorespiration from occurring. These cells, called bundle sheath cells, in these C4 plants prevent that photorespiration from occurring. And this is how they do that. During the night, when it's safe to open up the stomata and let CO2 in, they do that, and that CO2 is captured by this 3-carbon molecule called PEP. And so you have this 3-carbon molecule called PEP. It's 3 carbons. So I'll just put 3 here. And it's going to grab up a CO2, and that is going to make a 4-carbon molecule called oxaloacetate. Not that important, but to know that it's 4 carbons is important because that's where we get this C4. Sun comes out, stomatas close up, and now when the plant needs CO2, it has it stored up in these 4-carbon molecules. And so when the plant needs CO2, it just, as you can see, that 4-carbon molecule comes in, drops off a CO2, turns back to a 3-carbon molecule. That CO2 goes to the Calvin cycle, and normal can happen. This is plants like cotton, corn, plants that really love the sunshine, love the heat, and have adapted to that. Now there are other plants, desert plants, that even have a different adaptation. They're called CAM plants. CAM stands for crassulacean acid metabolism. Big words, lots of fun there. Here's what you need to know about that. Here's the C4 plant metabolism that we've already talked about, CO2, organic acid, Calvin cycle. In this place, with the CAM plants, what you have going on is no bundle sheath cells are necessary. You just have a different organic acid that is being used to store those CO2s at night so that during the day they can be released. And that acid is that crassulacean acid. Again, I'm not even going to write it because you don't need to spell it. You just know that CAM plants have a different C4 or different 4-carbon molecule that they use to store those CO2s.